We should move on to our next speaker, Professor Christina Smoke from Caltech. Let's welcome.
So today I'm going to focus my presentation really focusing on a particular area of research in my laboratory and discussing with you work that we've been doing in programming RNA information processing and control devices. So when we refer to devices in biological systems that are processing information and then sort of performing control operations, when we think about information in biological systems, we're really referring to sort of the information that's encoded in the properties of biological molecules. Molecules like proteins, nucleic acids, and also small molecules that are found in the cell. And so what we find when we go into organisms and try to reprogram their behavior, and this is also actually a limitation in sort of biological research in general, is that we're limited in our ability to access that sort of broadly information that's inside cells, to be able to transmit it into and out of cells non-invasively, and also to be able to act on that information. And so what that means then is that there's opportunities for us to really think about how to develop genetically encoded technologies that allow us to do that sort of information processing and control very broadly across different applications and different systems. And that's what I'm going to be speaking with you about today. So if we start very abstractly and just think about what are the functions that an information processing and control device has to perform, we can basically sort of distill it down into three basic functions that I'm showing here for a simple device. And so first, the device needs to be able to have a sensing function. This is going to allow it to sort of access and detect information that's inside of your biological system. The other sort of function that it needs to have is its actuator function, and this is going to allow the device to effect a response on the cell. And the third function, which is one of the more important functions, is it needs to have what I'm referring to here as a computation function, and that's really the function that sort of communicates between the sensor and the actuation function. So that when the sensor receives information, it takes that sort of information that comes in, performs some sort of processing event on it, and then translates that to the affected actuation event that you're regulating. And so if we think about the different types of applications that we're accessing in biological systems. Okay, hold on one second. My computer has frozen, I think. Great. Okay, so if we think very generally about the type of information that we want to be able to access in biological systems, there are many different types of information. Things like small molecules, proteins, nucleic acids. These are the biomolecules that I referred to. Also, we'd like to be able to access things like temperature and pH and ion concentration. And so this is just a very general list of different types of sort of information that's encoded in cells that we want to be able to access. And if we think then about the different types of ways that cells basically actuate, right, or affect different responses, there's a variety of different processes that we'd like to be able to tie into. Many of these actually link to gene expression regulatory pathways, so processes like transcription, translation, splicing, but there are other types of processes that sort of occur post-transcriptionally, so things like being able to regulate enzyme activity and complex formation. So this gets to more thinking about the substrate that we want to build our molecular devices out of. We'd like to be able to build out of substrates that will allow us to broadly access different types of molecular information within cells, but also be able to actuate through these different types of mechanisms. And finally, the next sort of challenge for us is that if we think about building a technology that's useful for a broad community, we don't want to be building sort of instances of devices, right, that are very specialized, but we want to think about whether or not biology will actually allow us to sort of develop general frameworks, right, for assembling these types of devices out of simpler functions. So if we build libraries of sensor functions and we build libraries of actuation functions, can we actually develop frameworks that allow us to piece them together so that from those frameworks we could build many instances of these types of devices? And those are going to be the challenges and some of the sort of key research that I share with you today. So we've thought a bit about this problem, and the way that we sort of approach it is thinking about how to build those sorts of devices out of RNA, nucleic acid molecules inside of cells. And we became inspired to think about this from the many examples and the many different types of functions that have sort of come out as examples of how biology uses RNA to both as a regulator or a control molecule, but also as a molecule that can process information inside of different organisms. So what I'm showing you here is an example of a type of RNA molecule that acts as an information process and control device inside of living systems. And so what you see here is the secondary structure of the two-dimensional sort of predicted structural conformation fold of this RNA molecule. And the important thing to note is that it's sort of, you can sort of outline two different domains that it is composed of. It has a sensor domain, which is referred to as an aptamer domain, and it has this actuator domain, which here is sort of outlined as an expression platform domain. And the idea then is that information comes into this aptamer or sensor domain and is transmitted to affect a response to this expression platform domain. And so the way that these molecules are used inside of cells is that they can actually adapt two different conformations that you see here. In the conformation on the left, this expression platform domain is inactive, and translation would be on in the cell. In the conformation on the right, the expression platform is active, and translation would be turned off. And so the idea is that the molecule can adapt both of these conformations, but it favors this conformation when the input ligand to the sensor is actually at high concentrations in the cell. So that allows us to be a dynamic control molecule, right? So as the sort of concentration of the input to this control device increases in the cell, it's going to shift to favor this conformation, and the expression of that protein will go from high levels to low levels. Now, some of the interesting things about these molecules is that they are actually used quite prevalently in different bacterial systems, and you oftentimes find them being used to regulate metabolism. So the inputs to these molecules are small molecule metabolites that are inside of the cell, and they're regulating enzymes that actually occur in these pathways. So they're using these for sort of dynamic embedded control and as a way to regulate flux and optimize energy usage through pathways. And so these are sort of very exciting examples of the sort of functional diversity that RNA can exhibit. And there's several reasons why these are exciting. The first is that you can imagine, so over 15 years ago, technologies were developed that allow us to go into the laboratory and de novo select for new RNA sensors, meaning that we can pick which sort of ligands and molecules that we would like to detect and go into the laboratory and through different types of selection procedures, generate sensors that will respond to those ligands. So you can imagine with that procedure, we have the ability to go in and sort of develop, or theoretically develop, libraries of RNA sensors to different molecules that we would like to detect. And then you can imagine that we might want to actually sort of take these biological molecules and swap out the RNA sensor that exists and put in a new RNA sensor so that we build a tailored control device for our system of interest. The challenge with this is that these types of biological systems have not been optimized or sort of designed with the engineering properties in mind. And specifically what I'm referring to here is that they're not built on sort of a modular framework where it's possible to sort of take this aptamer or RNA sensor sequence, remove it, and then place it with a new one, but still maintain function of the device. And this is likely not too surprising because it can be in this case that biology hasn't necessarily evolved these molecules to exhibit modularity. That's not sort of the property that they desire out of it. But as engineers, we would like to build that sort of capability into the system. And so what we need to do is sort of take a step back and propose or develop a new framework, a framework through which we can take these different types of basic functions that RNA can exhibit, but then assemble them into functioning devices. And so that's what my laboratory has been working on for several years. And so the idea, again, is that if we refer to the basic functions that we would like out of an information processing and control device, we would define these functions of sensing, of actuation, and computation. And we can sort of generally define different types of RNA molecules that would exhibit those types of functions. And we can, in fact, go into the laboratory and develop libraries of different RNA components that would exhibit these types of functions and group these into these general categories. So if we think about developing a composition framework for assembling an RNA device, what we're really thinking about then is basically a framework for taking these different basic functional components and being able to link them in a way that we will consistently get a reliably functioning device out of it. And so then we have to think a little bit further about the way information is actually going to flow through this device. And so again, what's going to happen is our input ligand is going to come into our sensor domain. It's going to be transmitted and processed through our computation element, and that is then going to affect a response in our actuator domain. The actuator is going to, in turn, basically regulate, in the cases that I'm going to talk about here, the expression or the levels of a protein or protein targets of interest for us. And that's going to be what our output is. And so one of the things you're going to note here, and this is a specific approach, is that what we're doing is we're taking these three distinct functions and separating them into distinct components. Another approach would be to say actually double code components. So encode a sensor that encode a domain that performs both sensing and computation. And there are examples of that in the literature. But we're making a very specific decision to actually encode each of these functions separately in a domain, and that's going to allow us to actually build modular frameworks out of this, as you'll see. Okay, so thinking more specifically about the physical linkages that occur between these components, that's what is sort of represented in this diagram here. And so, again, just to be very specific, our computation domain is going to be a domain that we can modify to actually achieve desired gate function. You'll see what I mean by that in the next couple slides. The second point about this framework is that you'll see here another important part of it is that we're defining these points of integration, which you see represented pictorially on this little schematic here. Depending on the sort of points of integration or the nodes that we use to combine these different types of components, we're going to get devices that can perform different types of information processes. And you'll see that again in the examples that I sort of draw out, so just keep that in mind. So before going into a few specific examples, I want to just sort of highlight the way that the devices that I'm going to share with you today actually work. Again, the way that we've done this is that the actual sort of specifics of what the sensor is going to be detecting and how the actuator is actually working should be able to be generalized. But for the examples that I show you today, we're going to use an actuator which encodes a catalytic RNA. So this is an RNA molecule that can self-cleave. And when we actually link it to our gene of interest that you see here, what we're doing is regulating gene expression through a cleavage event. So if the ribozyme is active, you would get cleavage, and gene expression should be inhibited, or our protein should be at low levels. When we inactivate the ribozyme, our protein should be at high levels. And so that's how our devices are going to work here today. Okay, so we had previously described single input RNA devices last year. And so I'm going to go through basically the properties of the single input RNA devices fairly quickly, but just to sort of cover the important properties of these devices. And the first sort of property that's important, as I highlighted in the previous slide, is this computation domain. And so what you see here is that we're going to be using basically competitive hybridization events between two strands, a green strand and a red strand, that are going to compete for binding to this section of the computation domain. And depending on which strand is bound, you're going to get a different sort of functional state of the device. The reason that this is sort of an important design aspect is that there are computational programs that allow us to predict the secondary structure, the two-dimensional folding properties of an RNA molecule. By using this type of mechanism, it allows us to rationally or forward design the types of devices that I'm going to show you here today. So this was the first type of device that we built, and we would call this an on-switch or a buffer gate. An on-switch is basically going to take our input concentration, which is shown here on the x-axis of a response curve, and translate that to an increase in the device response. And so first, let me just go over some specifics of the molecule that you see here. So we're still representing that secondary structure or two-dimensional structure of the molecule, but now I've inserted the nucleotides that are actually sort of represented within the switch. So the important thing is sort of to focus in on this region, which is the computation domain. And so what you basically see is that similar to the natural sort of RNA information processing and control devices, the molecule adapts two different conformations. In the conformation on the left, the sensor is not in a conformation that allows it to bind its input molecule, and the ribozyme is active. And that means gene expression should be low. In the conformation on the right, the sensor is correctly formed, and the ribozyme is inactive, and that means gene expression should be high. And as you would expect, as that input molecule increases inside the cell, it's going to shift the distribution to favor this conformation. And so we would expect a buffer gate activity, which is what we see here. A few important things about the response curve, just before going on, is that you'll notice that what we're reporting here, if you're looking closely, is the concentration of ligand to this RNA device, but it's a concentration that we're adding to cell culture. Okay? And actually, let me just take a step back and denote that these are experiments that were done in yeast. So we're encoding our system on DNA, we're putting the DNA into our yeast cells, and we're having the yeast make the device in the system. And then we're measuring an output, and here we're regulating a fluorescent reporter protein because it's easy for us to measure. Okay? So this is the concentration of the input molecule that we add to the media. For this particular input molecule, it has a hard time getting across the cell. So there's about a 1,000-fold drop across the membrane. So you should think about the intracellular concentration of the ligand being about 1,000-fold lower, and that matches the affinity of the aptamer that we're using. The other sort of important note is that what we're showing here is basically the difference between the on and the off state of the device that we're showing. This will become important as we start talking about programming the response curve in another few slides. The other sort of important point then just to make is that, as I said, we can program sort of the function of the device in this computation domain. So just by basically changing the sequence within the computation domain, making no other changes to the other domains of the molecule, the sensor and the actuator, we can now change the function of this device and change it to an inverter gate or an off switch. And so, and as you see here, this is the response curve of this particular device inside of yeast, where now we have sort of an off behavior with increasing ligand concentration. So, as I said, one of the things that we wanted to sort of, one of the important properties of these types of frameworks is modularity, because that will really sort of give us speed as we move towards making many instances of these devices for different applications. So we wanted to look at whether we have actually, in fact, developed a modular framework, and that would really be the ability, in this case that I'm going to show you here, to replace that sensor with other RNA sensors that might be of interest to us. So this is this type of schematic that we might outline for a type of sensor, direct sensor replacement strategy. We're drawing this little box around the original sensor that we had in our device. We can draw a box around a new sensor. So this would be a nucleic acid sequence that encodes a sensor that now responds to a different type of small molecule, tetracycline. And so we would just sort of directly replace the sequence. And if our system is modular, we would expect that we would still maintain the function of the device, which is a buffer gate, but now have it responsive to tetracycline. So we went ahead and sort of did these studies, and again, we're sort of showing you characterization study data in yeast. I'm representing the data slightly different here, okay? So let me just take a step back and explain. Now we're actually showing you the full device response. And so what we're showing here on the y-axis is actually the full transcriptional range of the promoter system that we're using. And what you see is sort of the device response across this transcriptional range. And so what you would note then is that there is a basal level, right, in the off state. There is gene expression level associated with that, right? That's above the background of the cell. And so, again, what we're showing here is now not the sort of response curve with the input concentration, but just the swing in the absence of ligand and the presence of ligand, as you see here. The important thing to note is that this is the data that I showed you previously for the theophylline responsive on switch. Okay, so this is how the data would be represented here. What you'll notice that when we replace it with the tetracycline aptamer, it still maintains on switch function. And the other important sort of thing to note is that it has the same sort of basal level. That's important because that basal level is actually not encoded within the sensor domain. It's encoded within the computation domain, and it again sort of supports the modularity of the system that we're building here. Okay, so I'm going to actually go on to the next, not focus on the rest of the data on this slide due to time. Now, sort of, as I had just said, these systems exhibit basal expression level. And that those sort of quantitative aspects of the response curve are programmed within the computation domain. So the computation domain is a part of the molecule that dictates the energetics through which this device switches between those two different states. And so what you can imagine then is because it's working through a competitive hybridization event, if we change the nucleotide sequences very slightly, which would change the predicted energy, we should actually be able to change the quantitative aspects of that response curve. And so just to show you that this is in fact the case, what I'm showing you here sort of, and it might be hard to sort of see, but I'm showing you here sort of a zoomed in region of the sequence of the computation domain. This was the original one for the original on switch that I showed you. And here we're making very small nucleotide changes across a series of different on switches. And what you see is that we are actually changing that output swing associated with a series of different on-switch devices. And so we can change it so that the device exhibits very low basal level like you see here. The important thing to note, and this, I'm not going to go very specifically into the energetics because this was previously reported, is that the predicted sort of response properties of the system match the energetics that are predicted through the folding programs. So if we change the sequence to sort of favor the ribosome active state of the device, we'll get devices with lower basal levels. So we are able to sort of tune the response properties of these switches. One thing that I want to sort of highlight here is that, because I am going through this fairly quickly, is that there is a poster presentation from a graduate student in my laboratory that actually focuses more on the quantitative aspects of tuning this sort of response curve that you see for these types of RNA devices. And so if you're interested in learning more about this, I would strongly suggest that you actually go speak with Chase Beisel and go to the poster presentation this afternoon, because there are very interesting aspects about sort of tuning the quantitative properties of the response curves. So now what I want to sort of focus on is some more recent work on how we actually basically took that framework that I described to you and that we had reported on last year and demonstrated that it could be extended, right, to actually make RNA devices that exhibit more complex and higher order types of information processing. So everything that I had showed you in the first part of my talk was single input RNA devices. And so now in the next set of slides, I'm going to sort of just show you examples of how we can basically take the sort of remaining points of integration that you see here and build different types of computation schemes with these RNA devices, but still using and just assembling these simple, well-characterized components that you see here. Okay, so the first type of extended framework that we described is one that we would refer to as signal integration within the 3' untranslated region of our transcript of interest. So here, essentially, what we're taking is we're taking single input devices, treating them as modules, and just linking them in series within the 3' UTR of our target gene. Depending on the functional properties of each of these single input gates, you'll be able to perform different types of computation. We've shown that you can build AND and NOR gates, also bandpass signal filters, and output signal filters. The reason, if you think about the way computation is being performed here, is that only one of these devices needs to be active or in the ribozyme active state to actually turn off gene expression, right? So that allows you to build these different types of computations from that sort of integration scheme. To sort of zoom in on a couple examples here, these are going to be sort of ways that we represent these high-order devices. So I just want to walk you through the representation that you see here. We're drawing boxed regions around the single input gates, okay? And then what we're showing, the triangles basically show the relationships between the input and the output from a single input gate. So this sort of demonstration of the triangles here, this would be encoding for a single input buffer gate or on switch. Gene expression increases as your input to that gate increases. So the type of device that we're showing you here is basically 2 buffer gates sort of linked in series. If we do that, what you would expect is that we would actually just shift the signal, right? Because both of those buffer gates would be acting in combination at the same time. And that's the data that you see here. So we're still representing the data the same way across the full transcriptional range of the system. What you see is 4 different single input gates that I previously showed you. So basically, 2 on switches to theophylline, one buffer gate to tetracycline, and actually 1 inverter gate to theophylline. Next to them is just the two of these gates in series. And what you see is that in each of these cases, the signal coming out of the device is shifted lower, which is what you would expect. The other important thing to note is that you could actually make a very simple prediction about where you'd expect the base level of the device to be from the performance of the single input gate. What I'm showing you here in gray circles is where the predicted sort of base level should be, and you see that in each case, the predicted level matches sort of the experimental level that we see. That's important because it confirms that each one of these devices is actually acting independently and don't exhibit crosstalk in this framework. The next thing to just sort of show as a demonstration is to sort of highlight the diversity of different types of computations that you can get with this framework is that if we now change it so we have 2 buffer gates that are linked in series but now respond to different inputs, you'd expect instead of getting a signal shift, you should see AND gate behavior, right? So that only in the presence of both inputs is gene expression turned on because both of these devices are then activated. And that's what you see there is if we actually characterize this device within yeast cells, now I'm just reporting the difference instead of across the full transcriptional range, that gene expression is only turned on when both inputs are present. We can make a very simple change to this device and now just encode 2 inverter gates, right? Instead of 2 buffer gates that respond to 2 different inputs, we would predict that we should now have NOR gate behavior, where gene expression is high only in the absence of both inputs, and that's what we see here. I don't have a lot of time, so I'll go through this quickly, but just to say that we can now actually build other types of extended frameworks. So instead of linking in series, we can actually link these sensor computation components off of 2 different points of integration with the ribozyme. This would actually allow us to access different types of computations, and in particular, we show NAND and OR gates. That's because computation is actually performed differently in this example, right? So in this example, both of these sort of internal gates that you see here have to be in the active state for the ribozyme in order for the ribozyme to be active. If either one inactivates it, that's going to dominate the sort of output of the system. So as we show here, that sort of computational integration would allow you to build a NAND gate device, right? Where if we link 2 different internal buffer gates, inverter gates, we would expect gene expression to be high unless both inputs are present, right? Because it'll be only in that case, when both inputs are present, that you activate the device. Or sort of you activate the ribozyme. And that's what we see here when we characterize the system inside of these cells. The final sort of framework that I just want to go over with you is one in which we're actually linking now these sensor computation elements in series, right, off of one stem of our actuator. Now, again, this is performing computation through a different way. Now we're performing computation sort of in series, right, across this chain of internal inverter gates, where the gate that's linked directly to this actuator is going to dominate the active state of the ribozyme. In doing this, we're able to show that we can build and OR gates and also systems that perform program cooperativity. So just to show you an example of this, I show you here, we can use this mechanism to build an alternative AND gate. So this is one where we're now linking two inverter gates, one responsive to input B, okay, and the one that's closest responsive to input A, which in this case is Theophylline. And so what you see is that input B has to first bind this internal gate that's sort of distal from the ribozyme. It changes the conformation to allow input A to bind the next internal gate, which will then inactivate the ribozyme. And that allows us to build an AND gate system, the data for which is what you see here. The final example that I want to show you, because it is a different type of computation, but also built upon this framework, is one in which we actually build program cooperativity or signal gain into our system. In this case, what we're doing is we're using two of these internal gates that are responsive to the same input linked in series to our device. And what we're actually doing is we're sort of tuning, we're using our ability to change the sequence of the computation domains, right, which link these elements, to change the energetics. And what that allows us to do is basically program the effective binding affinity of each one of these binding sites. And so what that allows us to do then is sort of program cooperativity or signal gain into the response of the system. And so what you see here is an example of one of the devices that we build. We measure the signal gain by measuring the Hill coefficient of our response curve. This is just an example of a device that does not exhibit program cooperativity. It has a Hill coefficient of 1. And this particular example here is a device that exhibits a Hill coefficient of about 1.7. What we saw is when we built a series of these is that the measured Hill coefficient roughly sort of correlated with the energy that we programmed into this step here. Okay, so I'm going to finish up in about 1 or 2 minutes. And what I had planned to do was just sort of touch upon a couple different application areas for these systems. And so I'm going to go through it fairly quickly. But the first one is just to say that one thing that we can begin to do with these systems is actually use them to non-invasively detect metabolite concentrations. And so in the example that I'm showing, so in all the examples that I've shown you thus far, we have been exogenously adding the input molecules to our cells of interest. And we wanted to sort of look at whether we're actually able to transmit information that's inside of cells, that's sort of endogenously present inside of cells. To sort of outside of cells. So this is an example where yeast are performing a very simple conversion of a substrate xanthocene to intracellular metabolite xanthine. Normally what we'd have to do to quantify and sort of look at production of xanthine over time is an invasive technique where we grow the cells at different time points. We make cell extracts. So we harvest the cells, we make cell extracts, and then we shoot those extracts onto an analytical instrument like an HPLC-MS that you see here. And the data that we would get from such an experiment shows us that if we feed xanthocene at time point 0, we don't start seeing xanthine accumulation in the cells into 24 hours, continues to 48 hours. Instead, what you can imagine doing is taking these RNA devices that I showed you, swapping out the sensor domain so that it instead recognizes xanthine, this internal metabolite, but still linking it to GFP fluorescence. And if we do the experiments here on the same cells but measure fluorescence at each of those time points, what you see is that at the time point where xanthine started accumulating in the cells, we see increases in GFP fluorescence, and it continues across the sort of state of growth that we saw here. I'm going to skip this slide due to time, and I just want to make one more note, but I'm going to not spend too much time on this application because we have a poster presentation on it where you can get more detail. But this is an application that we've been working on with collaborators at the City of Hope in regulating T-cell proliferation, so a cellular therapeutic application. So basically what I showed you, and I just want to highlight in this slide here, is that all the devices I showed you were devices that we had characterized initially and optimized inside of yeast. One of the nice things about the devices that we built is that they're portable, meaning that the mechanism that we chose to build these devices through actually translates across different systems. And in this particular example, it translates to mammalian cells. Okay, so we don't have to redesign the device to go into different systems. So what we can do is basically replace GFP with a functional gene in order to regulate function for different applications. And in this particular application, what we're looking at is to regulate the proliferation and activation of T cells. Okay? So we're regulating now, instead of a fluorescent reporter protein, a cytokine that will regulate T cell growth. So I'm not going to, again, as I said, spend too much detail here, just to show you that these response curves that I showed you in yeast translate to mammalian cells. Okay? We use the sort of combinatorial design principles that I showed you to lower the basal level. And what you see here now is that we have the same transfer functions or similar transfer functions, but instead of fluorescence, we're regulating relative percent viability. And this just shows you that we can sort of tune it to get to the set point that we want for this application. We're also now doing animal studies where we're taking T cells that we've engineered with these devices, putting them into mice, and basically showing that we can regulate growth across very long time periods in response to drug. So in the presence of the drug, we would want the T cells to proliferate, which is what you see here, and shown in an image here. And in the absence of animals that don't receive the drug, you would not expect to see T cells, which is what you see here. So again, if you're interested in the details of this work, you should go see a poster presentation by Yvonne Chen, which is a graduate student in my laboratory, who's doing this work, and you can get more details on it. But the point that I want to take away from this talk is that by building these frameworks, right, frameworks that have modularity and portability, it allows us to do a lot of the optimization in an organism that's easy to work with, like yeast, move it directly over to a sort of more difficult application, like cellular therapeutics, but still have it work and have already figured out the tuning principles, and then be able to use modularity such that once we develop RNA sensors to clinically relevant small molecules, we can swap them in and still maintain function of our devices. I will not go through this. I will thank the people who are responsible for the work that I shared with you today. In particular, all of the sort of foundational framework on the RNA devices that I shared with you today was done by a very talented graduate student in my laboratory, Mangyang Lin. I just touched upon Yvonne's work in the T-cell engineering project, but I definitely encourage you to go visit her poster for more details. Finally, I'll just sort of highlight, there's a presentation from my laboratory tomorrow that's on a very different aspect of the work that we do, metabolic engineering used for drug production. And so if you're interested in that sort of application area, you should definitely go to that talk as well. Thank you for your time.
Well, as it's pretty late, and we can take one or two short questions before we go out, take photo questions. Longer question we can leave for lunch discussion. So, any questions?
I have a small question. Have you ever tried to evolve a RiboSwitch to censor the in vivo product? For example, AHL, which is the product of LucasEye, and thus we can integrate the RiboSwitch to a more complex circuit.
Yes, that's a great question. And so the question is, can you integrate these types of devices or RiboSwitches to basically so that you're responding to endogenous signals inside of the cell? I showed very quickly at the end one example where we weren't linking it to function, but we were sensing a metabolite that was being produced by yeast, right? And we were just linking that to a fluorescent output signal. But you could imagine, and I think this gets sort of to some of the more interesting downstream applications of this area, right, is that if you think about where these systems are used naturally by biological systems, they're used to produce embedded dynamic control of metabolism, right, to produce feedback within metabolism so that the cell can basically respond to changes in precursors and intermediate metabolite levels and regulate enzymes in response to that, right? And that allows you to basically divert flux through different pathways and to optimize energy usage in response to sort of in-time conditions in the cell. And I think that's where some of the very interesting applications get into place. With a lot of the sort of application areas that we hear about, and a lot of different examples of people doing metabolic engineering, right? The tools that we use to sort of optimize those pathways, most of them are static, right? Control, right? Where you're just sort of constitutively expressing enzymes, especially when you go into scale up, because a lot of the inducer molecules that we use are quite expensive, right? And so you don't want to actually scale that typically very high. So another strategy, especially when you're doing sort of batch fermentations, right, is to start using these to implement sort of more sophisticated control schemes, thereby allowing you to sort of optimize energy usage and increase product in your pathway and minimize byproduct formation.
So last question.
What kind of range of protein concentrations can you control with RiboSwitches?
Okay, so what kind of range output is what you're asking, right? The sort of dynamic output swing?
Correct.
Yeah. So there's a couple ways to answer that. And the reason is that it somewhat depends on the organism that you work in, but also the actuator that you're using. So so it's hard to give a precise answer. So theoretically, the range that you could regulate would be the transcriptional range of the promoter system. However, what happens is that you run into some like system and practical limitations that sort of reduce that range as you go along. Because in order to access that full range, you'd really have to push the energetics to favor the off state. What that means is that you then have a much higher sort of ligand concentration that you need. So there becomes a trade-off in this, right? And so for those of you that are interested in this question, and I think it's actually really interesting and sort of important question, I would again encourage you to go see Chase Beisel's poster during the poster presentation, because he's done some computational work that really looks at that looks at like the theoretical sort of maximum and then how you would actually design towards that. What you saw in the presentation that I gave here is that for this particular type of device, one that works through ribozyme actuation, many of these, some of the ones that you saw, there's about a 50% dynamic range, right? But those are ones that have higher basal level, okay? So the ones that we sort of pushed the basal level to be lower had a percent dynamic range that was lower. However, we were also limited in how much ligand we could feed to the system and get into the cells. And what you saw, and I didn't point out, if we look at, say, theophylline and tetracycline, the dynamic range with tetracycline is much wider, and that's because it gets into the cells easier. Okay, so you have, again, these limitations as we're working with these model systems and sort of, how cytotoxic the ligand is that we're working with and how efficiently it gets into the cell. But as we start to select aptamers to new molecules, we'd want to be selecting for molecules that have more optimized properties.
Okay, thank you very much, Christina, for your fantastic talk. I will close the session by thanking all the three speakers for the session.